Good morning, everybody. My name is Mike Lawrence, and it's my pleasure to give this seminar today in honor of Joachim Frank's visit to the University of Cape Town. I'm going to talk in particular about the impact of cryo-electron microscopy on my particular field of structural biology, namely that of insulin and insulin receptor. So why are we interested in this system? Well, the obvious application is within the treatment of diabetes, both type 1 and type 2. Patients who suffer from these afflictions, typically in the case of type 1 diabetes, have insulin administered through almost their entire lives, and patients who have type 2 diabetes, certainly in the late stages of the disease, will have routine insulin injections. The problem with insulin therapy is that administration of insulin by injection is very far from ideal, in that the concentration of insulin that is achieved within the bloodstream does not mimic at all well that is found within a healthy patient, namely having a background basal level that increases in response to a meal intake and then declines again. Achieving that through injection is really quite hard. So with that in mind, various insulins have been developed over time, including long-acting insulins to maintain a basal background level, fast-acting to increase sugar uptake upon ingestion of a meal, and then the other issues as well that have been tried to be addressed, namely that of making orally bioavailable insulins, trying to make glucose-responsive insulins that will switch on in response to increased blood sugar concentration. And then the other issues regarding the thermostability of insulin and how it is stored, particularly for patients who are in warmer climates. So can structural biology tell us anything about this system that would be useful in developing better analogs? So what is the mechanism whereby insulin directs cells to take up glucose from the blood? This system is mediated by a receptor on the cell surface, the insulin receptor, which is shown over here in typical notation. It's a homodimeric disulfide linked receptor that has an exterior part that binds insulin, and then internal has two tyrosine kinase domains that transphosphorylate upon insulin binding to set up the signaling cascade that ultimately directs glucose uptake into the cell from the blood. Closely related to that is another system, the so-called insulin-like growth factor system, that will signal mitogenically into MAP kinase pathways in response to IGF-1 binding with the IGF-1 receptor. So these two receptors are homologous, as the name says, insulin-like growth factor receptor. And one of the issues is that there is some crosstalk between these two receptors, and I'll come on to that shortly, because it is the question of crosstalk is a major issue within the development of therapeutics that might tackle the system. Okay, so what about the structure of insulin itself? Insulin comprises two chains, an A chain and a B chain. These chains are cross-linked by two disulfide bonds, shown here in green, plus there is an internal disulfide bond within the A chain itself. Now, how these chains fold up is shown in this graphic here. The A chain has two helices, shown here in light blue. There's the internal disulfide within the A chain. The B chain has a single helix over here. There the disulfides that cross-link the two chains. And then on either end of the B chain, there is an extension N-terminally on the one side and C-terminally on the other. Now, through various analyses of sequences and mutagenic studies, it was deduced fairly early on that there are two distinct surfaces on insulin that are responsible for receptor engagement. It was also deduced that upon receptor engagement, this segment over here, the C-terminal segment of the B chain, must fold away from the core of the hormone to expose further hydrophobic residues within this region that in turn engage receptor. So all of this was deduced without any explicit knowledge of the receptor itself. Turning now to the theme of insulin analogs, if you look at the ones that are on the market, made by the various pharmaceutical companies that produce therapeutic insulins, you will see that almost all of them involve modifications to the B-chain C-terminal segment that I was talking about. That's 30 residues long. We talked about modifications to B28 and B29 or extensions onto the chain. Now, what these do in the case of fast-acting insulins is to try to destabilize that segment. In the case of long-acting insulins, they have various moieties attached to them that change the pharmacodynamics of insulin and its tissue distribution, and so hopefully enable it to be active in the system for longer. But if you think about that, almost none of that is actually exploiting any molecular detail regarding how insulin might bind to its receptor, which comes back to the premise of my talk. So we turn now to the question of the receptor itself. So the insulin receptor is a homodimeric receptor tyrosine kinase. The homodimer is formed by the cross-linking of two polypeptide chains by a pair of disulfide cross-links. The chains themselves are each broken into two segments, a so-called alpha chain and a beta chain, following furan cleavage at some point within the alpha chain. Most of the beta chain lies within the cell, so that's the intracellular segment that contains the tyrosine kinase domain. There's the transmembrane, juxtamembrane segments, and phosphorylation is believed to occur within the system. Within the extracellular portion, the ectodomain, there are a set of domains, the first and second leucine-rich repeat domains, a cysteine-rich region, and three type 3 fibronectin domains, FN3-1, FN3-2, and FN3-3. The second fibronectin domain actually spans the furan cleavage site, so it has an alpha chain component, a beta chain component, and it also, this cleavage site occurs within a big loop within the domain, the so-called insert domain ID. At the C-terminal end of the alpha chain is a segment called the alpha-CT segment, and that is absolutely critical in insulin binding, and I'll come back to that in a later slide. I'm now going to describe some early electron microscopy studies of the insulin receptor. Here on the left, we have holoreceptor reconstituted into lipid vesicles, and you can see that there were two classes of particles that were discerned. The one of which was a sort of T-shaped structure, shown over here and over here, and the second of which was this capital lambda-shaped particle or inverted V particle, shown over here. In a separate study by Tulloch et al. At CSIRO, undertaken when I was working at CSIRO, they discerned two classes of, or two views of particles, one of which was this, again, the V-shaped particle seen over here, and the other of which was this pair of parallel bars that they deduced with a top-down view of the V shape. And they came up with this sort of model shown here, which, although wrong in detail, was correct in other respects. And I'd point out that a UCT master's graduate from the Department of Applied Mathematics in Waddington contributed to this study. I turn now to a crystal structure that we published in 2006 of the receptor ectodomain. This was quite a remarkable structure determined in Colin Ward's laboratory. It was an enormous investment of CSIRO. They contributed about $20 million to this project over 15 years to obtain this structure. So it was no mean feat. But let me talk you through it. So as you can see straight away, it has the capital lambda shape that I talked about earlier. The membrane here is purely for purposes of illustration. And the two monomers assemble in a sort of anti-parallel fashion, twofold axis running down the center. What we deduce immediately from the structure is that on the opposite side of the membrane, the tyrosine kinase domains would be held apart by this shape, and in other words, would not allow transphosphorylation. So somehow upon insulin binding, the structure must close up, so to speak, and allow the tyrosine kinase domains to come together and to transphosphorylate. I want to draw your attention to one thing in particular, and that is where the alpha-CT segment ends up. So that sits atop the L1 domain, shown here in light cyan, of the opposite monomer, and there's the symmetric version on the other side, and this forms the primary insulin binding site, and I'll come back to that later. So whereas the V-shaped structure that I've just shown you reveals where on the surface of the receptor the primary binding site is located, it doesn't show at all how insulin might engage that site. And so when I moved to the Walter and Eliza Hall Institute in about 2007, I set up a laboratory to try to address that question. And the strategy we adopted was to make the smallest possible piece of the insulin receptor that would recapitulate the primary binding site, and then to complex that with insulin and to try to crystallize it. The result of those structures is shown here within this little video. So on the left, I've got the L1 domain of the receptor plus an exogenous alpha-CT peptide. On the right, I've got the closed conformation of insulin, as I showed you earlier. So in this video, you can see what happens. Firstly, the B chain of insulin folds out, as had been predicted. There's a reconfiguration of the alpha-CT segment and an intimate docking of the alpha-CT segment into the core of the hormone. So that was quite neat and explained an absolute wealth of the prior biochemical data. So what the previous two structures showed us were, on one hand, the structure of the APO receptor, on the other hand, the way in which insulin bound to its primary binding site. What we really wanted to know, what were the conformational changes that occurred in the receptor as a whole upon insulin binding that would bring about the juxtaposition of the tyrosine kinase domains themselves. And it was about this time that modern cryo-EM emerged. It offered us immediately significant advantages, the first of which was we could drastically cut our cost of protein production. The second of which was that it enabled us to deal with multiple stoichiometric classes simultaneously, and that was always a concern. We didn't know the stoichiometric mix of insulin and receptor that we had in our preparations. And the third of which was the whole question of crystallization is no longer an issue. So we latched onto this technology as rapidly as we could. Okay, so what did cryo-EM reveal? This was the first structure that was obtained, not in our laboratory, but in Bridget Carragher's laboratory, and published in 2018. And let me talk you through this. So here are two different views of the structure. What has happened here is that this is the insulin molecule bound to L1 and alpha-CT, shown in magenta in the background. The structure is twofold symmetric, as can be seen in this top-down view. There are the two insulins bound, looking down the twofold axis. What is absent from the structure are the legs of the receptor. So they are disordered in some way, but undoubtedly they will be closer together than in the structures of the APO receptor. But what has happened is that the insulin L1 complex has moved the L1 domain from down here, where it is attached to the leg of the V. It goes right up to the head of the receptor. So that's the first remarkable thing. The second remarkable thing is that the alpha-CT helix is much longer than we saw in our crystal structures. So there's a sort of supercoiling of that helix back down towards its linkage back into the alpha chain. The third point is that we now see insulin engaging the first of the fibronectin type 3 domains, the one in green over here, and there's this additional surface of insulin that binds to this receptor. So this was quite surprising, but there were a number of problems with this. The first of which is what is actually happening with the legs down here. The second of which is why is the stoichiometric arrangement 2-to-1? We were not expecting that because under physiological conditions and our understanding of the kinetics, we should only have a single insulin bound to the receptor. The third problem that arose from this structure was that although we now see insulin engaging another element of the receptor, the surface that involved was actually not what we had always believed to be the second receptor binding surface of insulin. So this was quite astounding, but it raised a whole set of questions. So at the same time, we were working on a different construct of the receptor that had been modified to permit high affinity insulin binding and to contain elements that would mimic membrane embedding. And the structure we obtained here is shown on the left. This was done in conjunction with Sanofi Aventis and with EMBL in Heidelberg. Let me just start the video, and this is what we obtained. You can see within the structure, the legs of the receptor are now together. There is only one insulin molecule bound to the receptor, and that the second binding site of insulin shown over here actually remains in about the same conformation that we see in the APO receptor. So in other words, this L1 domain is still in the sort of down conformation, whereas the other one has swung up into the up conformation. There's the bound insulin. And somehow this together brings about a conformational change that allows the legs to come together. Beyond that, the detail is very similar to that obtained in Bridget's laboratory. The insulin again engages with the adjacent FN3-1 domain, shown here in green. There is the extension of the alpha-C helix into a much longer conformation. But as I point out, the stoichiometry is now 1-to-1, which is what we would expect under physiological conditions. But still, the final conundrum is not solved, and that is, why do we not see the second receptor binding surface of insulin actually engaging the receptor, and we see, as it were, this third small interaction at that site? So I'm now going to show you a little video that summarizes what we've learned thus far. In the center here is the APO insulin receptor structure. On the left is insulin in its closed form. And if I start the video, you'll see what happens when they dock together. Here's insulin coming in. It engages L1 and alpha-CT. Those elements are then transported up to the head of the receptor. There's the additional winding of the alpha-CT element that occurs. The legs of the receptor are brought together, and we get this other binding surface that occurs up here at the top. So this is the 1-to-1 stoichiometric arrangement of insulin and receptor that we believe most closely mimics what is the physiological state of the insulin-bound receptor. This image is now a zoom in on insulin as it binds to the receptor surfaces that I've shown you. Here you can see the L1 domain, the first fibronectin type 3 domain of the opposite monomer, and the receptor alpha-CT helix. Insulin now engages those elements as follows. The B-chain helix sits on top of L1. The B-chain C-terminal segment is folded out away from the hormone core in the background there. Alpha-CT engages with the core of the hormone. And then we also see these additional interactions that have been highlighted over here that show engagement between a surface of insulin and the first fibronectin type 3 domain of the opposite monomer. And as I pointed out now on 2 or 3 occasions, is that from what is known from insulin binding studies, this surface is not actually the second binding surface of the receptor that had been prior discerned. That surface is actually somewhere over here and is not seen to engage receptor at all within these structures. So that's our remaining conundrum that still needs to be addressed. So the big surprise was then this structure obtained by the Koskan laboratory in Dresden in Germany. It is a 4-to-1 insulin to receptor complex. Within the structure we see 2 insulins bound at the top, each bound in fashion similar to that seen in our structure and in the Carriger structure, but the surprise are these 2 insulins bound down the bottom. And the intriguing thing about this is that the binding surface of insulin here to the receptor, which is to the first of the three fibronectin type 3 domains, that binding surface of insulin involves residues that were long surmised to be the second receptor binding surface of insulin. So even though this was obtained under non-physiological saturating conditions, it seems to contain within it elements that mimic in some fashion physiological insulin binding. And what has since been concluded is that what is happening here is that it is this interaction here that is relatively weak that actually initiates the binding of this insulin, which is the high affinity interaction. So binding here actually frees up this domain to enable it to capture further insulin molecule that will bind tightly to the receptor and affect the overall structural transition that occurs in insulin. I can just start the video so you can get a better view of how that is assembled. You can see the 4 insulins bound, but as I stress, this was obtained under saturating conditions. I think it's about a 20-to-1 insulin to receptor complex. So what have these structures shown us? Well, the first thing is that they've shown us the conformational transitions that occur in the receptor upon insulin binding. They show us how insulin engages its receptor. They show us the binding surfaces of insulin that are involved, and concomitantly explain the vast wealth of biochemical data and mutational data that have accumulated over decades regarding insulin binding. The question that has arisen and still needs to be adequately resolved is, do any of the higher stoichiometric complexes occur under physiological conditions? And that's the subject of quite a bit of debate. It is my view that given the fact that the insulin concentration in serum is picomolar, it is unlikely that we're going to get more than one of the high affinity sites occupied, given the fact that receptor binding is known to be negatively cooperative. Of course, all of the kinetic analyses have been carried out under in vitro conditions, and it is certainly possible that under physiological conditions there could be microenvironmental effects that alter the local insulin concentration and could, in principle, lead to the possibility that some of these higher stoichiometric states exist, but that would be the subject of further investigation. So then, just to allude very briefly to applications of the study, I've been involved in 2, one of which is a collaboration with the NHS laboratory now at Stanford in the design of very fast-acting insulins based on marine snail venom insulins. These venoms are used to capture fish and induce hyperglycemic shock into the fish, and obviously you have to act quickly before the fish swims away. And some of those principles can be translated into human analogs. The second of which is to insert a glucose sensing switch into the B chain of insulin so that it remains tethered to the hormone in the absence of glucose, and then in high glucose concentration allows release of that chain. And you can clearly see what the applications of that would be in diabetes therapy. So I'm not going to go into detail about those, but we have cryo-EM structures that have helped within the design of those insulin analogs. Some of that will still yet to be published. Finally, it remains for me to thank the many people who've been involved in these studies over decades, but I'd like to pay particular tribute to 3 people who have acted as mentors for me over my career. In particular, Peter Coleman, Colin Ward, and indeed Trevor Sewell, who I had the privilege of meeting here way back in 1980, I think, Trevor, after your return from Birkbeck. And you have certainly always been an inspiration for me. So thank you for your attention. This talk is now open for questions.